Okay, so I always get this question, what do we study for mole? Do we study only the PowerPoint? Do we study from the book? What do we study? The PowerPoint has all the material that you are responsible to study, okay? So I'm not gonna ask you any information which is not in the PowerPoint. But this doesn't mean that you should only study the PowerPoint. You should start by studying the PowerPoint, and then you need to practice questions, okay? And you need to refer to the book if you don't understand the information in the PowerPoint. If you want to read further about the material explained in the PowerPoint, you go back to the book. But the PowerPoint alone will not be enough because you need to practice questions. And in the PowerPoint, I will put 1 or 2 questions. You need to solve problems, and these should be accessible to you either from the previous batch question bank or from the book, because the book has few questions and it has an access code for more questions, or from other resources such as, I will show you at the end, there is a flashcards book, a companion book for your textbook, for your Lipping Code textbook. There is a companion book that you can use, and I'll show it to you at the end. It's actually shown in the last slide. Clear? So yes, you need to study the PowerPoint. The PowerPoint has all the knowledge that you need to study, but it's not enough to secure an A. To secure an A, you have to practice questions. Clear? Good. So while this is opening, which is taking forever, I will start talking about amino acids. Can you all hear me? Yes? Okay, if you not hearing me properly, just raise your hand. I will try to adjust my voice. Am I too loud or? Great. Okay, we have to get used to this place because normally we don't teach here. Normally we teach you in the female and the male lecture theater. We're not used to teaching here. This venue is only used for the TBL. We don't normally use it for teaching, so it's a new experience for us and for you. Today we're going to start talking about amino acids or amino acids, depending on which accent you want to use. And I'm sure many of you have heard of amino acids. What are amino acids? They are the building blocks. The building blocks of what? Yes, they are the building blocks of proteins. Okay, so in our body we have large molecules, and we have 4 different types of large molecules. What are these 4 different types of large molecules? Carbohydrates, lipids, proteins, nucleic acids. Okay, so these are the large molecules in our body, and in biochemistry we're going to study them and their pathway in details, and we're going to start with the proteins. And the proteins are made from monomers, repeating units of a building block, and the building block is an amino acid. Many students, especially from Arabic speaking school, they confuse nucleic acid with amino acids. No, it should be crystal clear to you as a biochemistry student that nucleic acid, it's DNA and RNA. This is the genetic material. Okay? So whenever you hear the word nucleic, think of the nucleus, and in the nucleus we have the genetic information, right? When you hear the word amino acid, you should immediately think of proteins. Can you give me an example of a protein? Enzymes are protein. Actually, enzymes are one of the most important proteins in our body. So the enzymes that help us with the digestion, the enzymes like, yes, amylase, protease, lipase, all these enzymes that help us with digestion. They are one type of enzyme, but also we have the enzymes that make all the reaction in our body happen, such as, can you give me an example of an enzyme? Amylase for digestion, something not for digestion, an enzyme? Okay, kinase. So all these enzymes which we'll see in biochemistry, they are proteins. So proteins can be enzymes. What else? Can you give me other examples of proteins? Collagen, keratin, insulin. So proteins have different functions in our body. Actually, proteins, they are the most abundant large molecule in our body. So the most abundant large molecule in our body is protein. That's why you need to understand what are proteins and what they are made from. So we'll start today by talking about the building blocks, the monomer, the one unit of the protein, which is the amino acid. And then we're going to talk about proteins in more detail. Okay, so the amino acids that make up our proteins are how many? How many amino acids make up our protein? 20. We call them the 20 standard amino acids. Do we have other amino acids which are not part of proteins? Or all amino acids, they just exist in our proteins? No, we have other amino acids which don't make up the protein, but the one that make up our protein, we call them the standard amino acid, and this is going to be the focus of our lecture today if I manage to open the lecture, because at the moment it seems that I'm not able to open it, network error. Sorry? Arrow? Resume, okay, thank you. Opening when complete, okay, take your time. Okay, so we're gonna learn today about the 20 amino acids. These are the ones that make up the proteins in our body. We call them the standard amino acids. Can you give me an example of an amino acid? Histidine, lysine, leucine. Okay, so these are the 20 amino acids. So meet your 20 new friends. The 20 amino acids, they will accompany you not only in mole 1, but also in mole 2. So when you meet a new person, you have to at least remember the name, right? And maybe the nickname, so you need to know the abbreviation. I'm not gonna ask you to draw these amino acids, but you should be able to recognize what is special about each one of them. Okay, so, and you should be able to classify them. So this is the information that you should know about these 20 amino acids. Their names, their abbreviation, and what makes each one special, and of course, what is common among all of them. So what makes an amino acid an amino acid? Yes. Yes, the classification, yes. Someone also had the, yes, please? Okay, so you're answering the question. They all have a side chain, but this is not why we call them amino acid. Why we call them amino acid? What makes an amino acid an amino acid? Yes? They have amino group, yes. What else? Carboxylic acid group. They are attached to the central carbon, and they have an R and an H. So this is common among all the 20 amino acids. Okay, can you clearly see the diagram here? Yes, at the back, can you see my diagram? Yes, okay. So we have a carboxylic acid group, an amino group, a side chain, and an H. So all amino acids will have this structure. And what makes them different? The R. So the R will be different for the different 20 amino acids. Clear? Okay, so what do we call the central carbon? Yes, we call it alpha. Why we call it alpha? It's just a numbering system for carboxylic acid. So when we number any carboxylic acid derivative, we start counting after the carboxyl acid group, we start numbering this carbon, and this is going to be the alpha carbon. That's why we call it alpha carbon. And to this alpha carbon, we have 4 things attached: the carboxyl group, the amino group, the side chain, and the H. And because these 4 things are different, or when they are different, this will make the carbon or the alpha carbon what? Chiral. Thank you. And what does chiral mean? Okay, chiral or asymmetric. So it means it's bonded to 4 different things. Therefore, it can have different what? It will have stereoisomers. Okay? Yes, so what are, first of all, what are isomers? So what are isomers from your organic chemistry? Yes? Yes, so an isomer, so when we are defining 2 isomers, so they are 2 molecules that they have the same chemical formula, but they differ in their structure, okay? If they're different in the structure, like here, because of different bonds, this is structural isomer, we're not interested in this. Here, we are interested in stereoisomers. So what are stereoisomers? They have same chemical formula, but different 3D structure, okay? And this 3D structure can be in the type of what we call optical isomer or stereoisomer. So here they are mirror image, right? So this is what we define as, why we need to define this alpha carbon as chiral or asymmetric, because you need to understand that amino acids exist as stereoisomers or non-superimposable mirror image. So we have 2 configurations. We have the D and the L amino acid. Why you need to know about this D and L? So the D and the L, as I said, they are stereoisomers or non-superimposable mirror image. They have the same chemical formula, but a different 3D structure. And the one that exists in our proteins, the amino acid that make up our proteins, happens to be only the L form. So if someone asks you, do we have D amino acid or L amino acids? What are this D and L? D and L, they are 2 different configurations of the same molecule. And the one that exists in proteins are the L, as simply as that. You're gonna see stereoisomers again when you study carbohydrates, but this is not organic chemistry, this is biochemistry, so you don't need to worry a lot about it. For those of you who don't like geometry and organic chemistry, don't worry. Just remember that in our body we have L amino acids. Do we have D amino acids in nature? Yes. They exist in some bacterial cell wall, in antibiotics, but not in our body. In our body, we have only L amino acid. The type of stereoisomers that are present for this amino acid is the L type. If you want to find out what is the difference between the D and the L, you can read this, and then they will tell you which configuration is the D, which configuration is the L one. Okay, so let's repeat what we have learned so far. We've learned that all amino acids have this basic structure of alpha carbon, to which we have an amino group, a carboxyl group attached, a hydrogen group, and an R group or a side chain, which is different for the 20 different amino acids. When we look at these acid and base group, because carboxylic acid, of course, it's an acidic group, and amino group, it's a basic group, this will make us think about what type of species, when you add this amino acid to an aqueous solution, such as blood, what form will you get? If you have a free amino acid, it's going to ionize, and it will generate species. What type of ions? You will have this type of ions. At physiological pH, which is about 7.4, you're gonna have the carboxyl group donating its H, and the amino group accepting the H. Therefore, you're going to have in the same molecule a positive and a negative charge. And this is what we call in chemistry, what? Zwitterion. Okay, so it's an ion that has a positive and a negative charge. So this is the dominant species of amino acids, of free amino acid at physiological pH. What about the charge of the R? What do you think? Is it always uncharged? Yalla, who's clever and who took it in other courses or something, or who have read about it? We said at physiological pH, if you have a free amino acid, not part of a protein, a free amino acid is going to have a negative charge on the carboxyl group and a positive charge on the amino group. What about the charge of the R? What will be the charge on the R? Yes. Neutral? No. Yes. Negative? No. Okay. So, okay, you're getting there, yes? No, the charge of the R is independent of the charge of the carboxyl and the amino group, yes? Yes? No, you with the glasses, yes? Negative? Yes? Both at the same time? Yes, this is the correct answer. It depends on what groups we have on the R. Some amino acids will be positive, some will be negative, and some will be neutral, depending on what is the R. Actually, you should know that if you have this amino acid part of a protein, the property of the R is the most important, because the carboxyl group and the amino group within the protein are going to be busy. Busy doing what? Yes? Excellent. So all the internal carboxyl group and amino group will be busy in a peptide bond. Only there will be a free end with the carboxyl group and a free end with the amino group, and all the others will be involved in the peptide bond. What is the peptide bond? Yeah, it's the bond that add amino acids together to form a protein. So all the peptide bonds are going to be involving the carboxyl group and the amino group, so they're going to be busy. The only free group is going to be the R, and that's why we need to know the property of the R, because it will tell us what different charges we have within the protein and where is this amino acid is going to be placed in this protein, whether it's going to be outside, inside, etc. And what is the folding or the shape of this protein. So the folding of the protein will depend on the R. Okay, so this is regarding the charges. So at physiological pH, the carboxyl group will have a negative charge, the amino group will have a positive charge, and the R group will be variant. It will be different for the different amino acid depending on which group we have here. So these are your 20 friends, their full name and their nickname or their abbreviation. We have here, my favorite name is Serene, okay? This is not a name of a girl. This is serine, an amino acid. Okay, so make sure you know your 20 friends because you will meet them again and again in biochemistry. There is a disease, very serious disease called sickle cell anemia, which is caused by a substitution. There is a mutation that replaces one amino acid with another. What is the amino acid that is mutated in? So valine is used to replace Glutamic acid, yes. So glutamic acid, or GLU, is replaced by valine. It's very important that you read the abbreviation because you might get a question, or remember the abbreviation, you might get a question where you have GLN, and you need to know that GLN is glutamine, not glutamic acid, okay? So some of them, like glutamic acid and glutamine, they have the first three letters are common, so you have to know the abbreviation. Similarly, aspartic acid and asparagine. So usually when you have an N, it is the one that ends with glutamine. Okay, so make sure you familiarize yourself not only with the names, but also with the abbreviation. So I could write to you a peptide structure, and you will have the names like this as an abbreviation, not necessarily as the full name. So when you look at something like this, you should be able to recognize that here is the alpha carbon, right? This is the H, this is the amino group, this is the carboxyl group, and in red, what do we have? The different reds, they indicate here what? They are. So you should be able to familiarize yourself with an amino acid when they are free, alone, or when they are in the peptide bond. So you could have a question where you have to indicate where is the peptide bond here. Okay, so later we will revisit the concept, but you should be able now to have an eye for this. So you should be able to point this out as the peptide bond because it's the one linking the carboxyl with the amino, as we will see later. Okay, in biochemistry we have essential and non-essential molecules. But doesn't mean that essential means they are important and the non-essential they are non-important. So essential in biochemistry and in nutrition in general has to do with your diet. So what does essential molecule mean in nutrition? It's not produced by the body, therefore, where do you get them from? From the diet. Okay, always I tell my students to remember essential to eat. You have to eat them because if you don't eat them, you're not going to get them. You're going to be deficient in them because your body cannot make them. And when we study in Mol 2 the synthesis of amino acid, how we make amino acid in the body, we're going to study the synthesis of non-essential because the essential amino acids will not be made in our body. Do we need to remember them? Yes, because later, as a future doctor, if you have a patient with, a problem with the metabolism and they have a certain genetic disorder and they have a deficient enzyme in the pathway of one of the amino acid, you should be able to differentiate between essential and non-essential amino acid, whether you have to replace this amino acid in the diet or no. So that's why you need to have a concept about essential and non-essential. There is a mnemonic. I always try to use mnemonic in biochemistry to remind you of difficult or like multiple things to remember. Here we have PVT Tim Hall to remember the 10 essential amino acids, the ones that cannot be made in the body and you have to obtain them from the diet. Arginine, some books refer to it as semi-essential because it's only necessary during puberty and development. So during period of active growth, when the body is actively growing, this is when you need arginine. Later in adulthood, it will be whatever you make in the body, it will be enough. You don't need to get extra arginine from the diet. So this is regarding the definition of essential amino acids. Okay, so the importance of amino acids comes from its property as building blocks of proteins, as we have learned so far, and we define already what makes an amino acid. An amino acid is the carboxyl group, the amino group, so linked to the alpha carbon. This is what we have studied so far. So now we also know that why we call it alpha, because the terminology used to name or number the carbons in carboxylic acid group derivatives is to number carbon number 2, the one after the carboxyl acid group will be alpha, and that's it. Now we're going to study what is special about each amino acid, and we're going to start with proline. Why proline? What is special about proline? This is the structure of proline. So this is the alpha carbon, this is the carboxyl group, this is the H, and this is the R. So you can see it here again. So what is special about proline if you compare it, let's say, to alanine? Yes? Yes. So the R group is linked not only to the alpha carbon, but also to the amino group. And that makes proline a secondary amino group, because the amino group is linked to 2 carbon. So we call it secondary amino group, or sometimes they refer to it as amino acid. Okay, so the side chain is curving back from the alpha carbon to have a bond with the amine nitrogen. Okay, so just some terminology, we call it secondary amino acid or amino acid because of this unusual bond between the R and the amino group. Okay, but that unusual bond is going to make proline bulky. Okay, so proline is, because of its bulky structure, it's going to be useful when making structural proteins such as collagen. So you're going to find lots of proline in collagen because it has this bulky structure. But proline, because it's bulky, if it is present in what we call the alpha helical structure, it's not going to be very welcome there. It's going to interrupt the alpha helices because of its bulky structure. So yes, proline is good for the collagen because it's bulky, but it's going to be interrupting the alpha helix in globular protein, as we will learn later. Don't worry if you don't get this notion now, because we come back to this later. Okay, and here again is proline and a modified version of proline in the collagen. So we need proline, we need hydroxyproline and collagen because of their bulky structure, which cause this twisting of the collagen helix. So we're done with the bulky proline. Next, we have the branched chain amino acids. Okay, and here I have a baby formula. And this baby formula is free of this 3 branched chain amino acids, which are valine, leucine, and isoleucine. So why we call them branched chain amino acid? Yeah, as the name indicates, if you look at the side chain, the R in green here, it has a branch. 3 of them have those branches, so 3 branched chain amino acids, and the 3 of them need a special enzyme to be broken down in our body, and this enzyme is missing in an inherited disease where the children don't have a proper enzyme to metabolize or break down these amino acids, and the children will have this disease called maple syrup urine disease. Why we call it maple syrup urine disease? Maple syrup urine disease, what's this weird combination? Yes? Color of the urine? Yes? Yes. So these babies who have a deficiency in the enzyme to break down those branched chain amino acids, they will have a urine that smells like maple syrup. Now you're going to hate maple syrup. Sorry, no more maple syrup on your pancake. Use honey, honey is better. Okay, so MSUD, we will study it in Mol 2 in spring, inshallah. So MSUD is an abbreviation for maple syrup urine disease. It's a disease where there is a deficiency in the enzyme needed to break down those 3 amino acids. So these babies should be fed a formula milk that doesn't contain these 3 amino acids. Yes? The structure? No. But you should know that valine, leucine, isoleucine are branched chain amino acids. Again, as I explained at the beginning, you should know the name, the abbreviation, the classification, and what is special about each one of them without being able to actually draw the structure. Yes? This is genetics, and still in spring you're going to study genetics, and you're going to have Dr. Hasnan talking about all the metabolic disease, and you can ask him all these questions. It's too early now. We cannot learn all the knowledge in one day, so we have to learn things one thing at a time. I can give you a lecture here about the metabolism of these amino acids, but it's too early. Now we need to know what are amino acids and what is special about each amino acid. So let's focus. It's good to think outside the box, but we cannot address all these questions. Yes? I can't hear you, sorry. Are they what? In the mother milk. Good question. I don't know. Again, now we're thinking about treatment of patients with MSUD, so that's very advanced. He's talking about genetics, you're talking about treatment. You know, in medicine, in year 1, you study the basic science. In year 2, you start understanding pathology a little bit, like, you know, pharmacology, microbiology, but it's, again, basic science. It's only till year 3 that you start talking about disease, the diagnosis, the treatment. Okay, so you can't learn everything in one day. So you have to take it one step at a time. Okay, so when we talk about amino acids, there are some modification to these amino acids. So amino acids, we can add OH to them, and this is hydroxylation. That's why we saw hydroxyproline. So this is one type of modification. We play around with them, so they get modified. So they are made as the amino acid that we know, but they can be modified after synthesis. So we call it post-translational modification. So one type is the hydroxylation, another type is the glycosylation. So now, when I say hydroxylation, what does it mean? You add OH. What about glycosylation? What do you add? You add sugar. Okay, so if you have a protein, let's say, and this protein contains an asparagine, which is an amino acid with an amino group here in the R. So this asparagine can be modified by adding a sugar to it. And this sugar, we can attach it to the N of the asparagine. That's why we call it N-linked glycosylation. Okay, so in biochemistry, we have N-glycosylation and O-glycosylation. So you're adding the sugar either to the N, and you add the N usually to asparagine, or you add the sugar to O, or the OH group, and the O could be either in serine, again, not serine Abdel-Noor, or threonine. Okay, so this is what makes these 3 amino acids special. Asparagine for N-glycosylation, adding sugar to its nitrogen, and serine and threonine for adding sugar to its oxygen on the OH group. So this is what makes these 3, asparagine, serine, and threonine, special. Now we're going to talk about the one that contains sulfur. We have only 2 amino acids with a sulfur group. And where is the sulfur group? In which part? In the side chain, of course, because, you know, we don't have S in all amino acids. We only have it in 2 cases, so it has to be in the side chain. And these 2 amino acids are the methionine. What is special about methionine also? You learned from Dr. Ahmad. What's AUG? Start codon for methionine. Yes, so methionine is the amino acid that is coded by the start codon. Another amino acid that contains sulfur is cysteine. But cysteine is more important than methionine. Why? Okay, methionine is important for the start codon bit, but cysteine, the sulfur group of it, is important because it has a sulfur at the end. It's actually SH. It's self-hydryl group. So like OH is a hydroxyl group, SH is a self-hydryl group. And this SH can interact with another SH to form a special type of covalent bond called what? Disulfide bridge. So SH react with SH, forming a special type of covalent bond, forming what we call a disulfide bridge. Okay, so this interaction will influence the folding of the protein. We're going to learn about protein folding next week and how it is influenced by different type of bonds. So disulfide bridge, so if you have two adjacent, two cysteine facing each other in the protein, they can bind to each other, forming a very stable structure because, you know, covalent bonds are very stable, and these are the disulfide bridges. So we learned about proline, the branched chain, the sulfur containing. Now we have the aromatic amino acid. What does aromatic mean in chemistry? Okay, so aromatic means they contain a benzene ring here. That's what makes these three amino acids. We call them aromatic amino acids. So we have three amino acids which are aromatic. These are phenylalanine, tryptophan, and tyrosine. Actually, if you look at them, two of them, they look exactly like each other with a small difference. Which two? Look at them. Which two are exactly the same except for an OH? Yes, phenylalanine and tyrosine, they are exactly the same except for the OH. Okay, so actually tyrosine is made from phenylalanine just by adding OH to it. So you should have a good, what they call it, examination, not examination. دقة الملاحظة. My English is bad now. Observational skills, yes, observational skills. So these three amino acids are the aromatic. Why we need to know about them and their bulky benzene ring? All amino acids help in protein synthesis. No, they help us to quantify. If you want to measure the concentration of a protein, you can measure it with the help of this device called spectrophotometer. Why? Because the amino acids such as phenylalanine, tryptophan, tyrosine, or the aromatic amino acid, they can absorb UV light at a certain wavelength. So if you put a sample with your protein in this device and you expose your sample to an ultraviolet light at a certain wavelength, you can calculate the concentration of this protein sample. And the device is called spectrophotometer. I think it was introduced to you in the previous lecture, right, for the lab? Did I show you the instruments? There should be, or maybe in the next lab, they will show you a presentation with all the instruments that you should see in a biochemistry lab, and spectrophotometer is one of them. Okay, so we talked about the special or the unique properties of each amino acid. Now we're going to talk about their clinical significance. The first and the common significance among all of them, and they make up our protein, right? But there are some amino acids, not only they make up our protein, but they are used to make important molecules in our body. And these are just examples. I'm sure you all know what is histamine, right? How many of you have allergies and they take antihistamine? Yes, we all It's very common, especially if you live in Riyadh, to have allergies and you need to take antihistamine. So antihistamine, it's a drug that works against histamine, which is a substance that is triggered by allergic reaction. And this histamine, it turns out, it's a derivative of histidine. So histamine, that is responsible for all the nasty reaction that you experience when you have allergic reaction, comes from the amino acid in our body, histidine. You just remove a carboxyl group here from histidine and you end up with histamine. Also, our thyroid gland produces an important hormone called thyroxine. Right? You all know the thyroid gland, right? And some people have a problem with the thyroid and they have to take the hormone thyroxine on a regular basis. So this thyroxine hormone is actually made from the amino acid tyrosine, but not by a simple procedure. No, you have 9 reactions before you make thyroxine from tyrosine. Also, important molecules such as the neurotransmitter that control our mood and how do we feel, such as dopamine. And actually here we have L-DOPA. What is L-DOPA? L-DOPA is dihydroxyphenylalanine. So we said that from phenylalanine to tyrosine you add one OH. If you add another OH, you will end up with L-DOPA, which is a medicine that is used to treat patients with Alzheimer's. So not only amino acids are needed to make the proteins in our body, but certain amino acids can have many clinical significance. So some of them are used to make hormones, some of them they make molecules that trigger allergic reactions, some of them they make neurotransmitters or chemicals that affect our mood. This is another example here. So not only I mentioned that L-DOPA is made from phenylalanine, but if you continue the reactions here from L-DOPA, you can get dopamine, which is also very important for our mood, but also you can convert dopamine to epinephrine. What is epinephrine? It's adrenaline. So adrenaline, the important hormone that regulates our fight or flight mechanism, is actually coming from the amino acid phenylalanine. Do we need to study this reaction? A question that I get every year. No, just remember that phenylalanine can be converted to several important molecules such as L-DOPA, DOPA, and epinephrine, without remembering the reaction at this stage, nor remembering the steps of the reaction or the structures. Another example of the clinical significance is the formation of serotonin. Serotonin is actually derived from the amino acid tryptophan. So we have 3 aromatic. We have the phenylalanine that is used to make tyrosine, and the third aromatic is tryptophan, and tryptophan is used to make serotonin. Does anyone know what is serotonin? Yes? It's the happiness hormone. What do we give to people with depression? SSRI, mashallah, very advanced knowledge. What is an SSRI? What does it do, the SSRI? It raises the level of what? Serotonin or serotonin, okay? So you don't need to know this information, don't worry. At this stage, just remember that tryptophan is used in our body to make the happiness hormone serotonin, and persons with depression are treated by drugs that raise their serotonin level. And actually, another approach or alternative medicine approach is to have a diet rich with tryptophan Rich food. Can you give me an example of something in your diet that is rich in tryptophan? Chocolate? No. Okay, that's your homework for next time. Search for which food ingredient, not now, not Google now, no. Remember Dr. Abdul Jabbar, what he said yesterday, if you're studying, you should keep your phone aside. So the same apply to the class. If you're in the class, put your phone aside. You're not here at a conference. We are in a lecture. Yes? Nuts and oats and turkey. Yes, turkey. Yes, turkey is very rich in tryptophan. So go and have a Subway now for your lunch. Unfortunately, you can't go yet for your lunch. We have still to finish. We started late, remember? Yes, I'm so sorry. I have to finish the last part because it's going to be a while. I don't think who's going to give you the next lecture is Dr. Abdul Jabbar. And my next lecture is going to be after the foundation, so you'd have forgotten what we took now. So I have to continue. Sorry? Mole exams. Did you read the email I sent to you? Read the email. You will know when is the mole exam. By the way, these lectures are not included in your foundation final, okay? I've told you before, the parallel courses lecture are not included in your block final. So mole, you don't study them for the fun final. They have their own midterms, their own final. Okay, let us continue. So as I said before, the carboxyl group and the amino group, as you can see them here, the carboxyl group and the amino group, they are busy in the peptide bond. That's why the R, here is the R for this phenylalanine, and this is the R for leucine. They are the one that dictate the property of the amino acid. They are going to influence the folding, and they are going to influence the properties of the protein in general. So this is just an example to show you that it is the R that is going to be interacting, and this is the R that is going to influence the folding of the protein. And it is the R that will tell us whether this protein or this amino acid, is going to be on the surface or inside. So if you look here at the circles are non-polar amino acids. So because they are non-polar, they have to hide inside the protein if it is present in an aqueous environment. So this is a soluble protein. In aqueous environment, it's going to hide away all the circles, and all the circles are the non-polar, the hydrophobic amino acid. Whereas if it is a transmembrane protein, a protein sitting on the plasma membrane, then all the non-polar amino acid can afford to sit on the what? On the surface, because the surface here is hydrophobic. So it's friend with these amino acids which are also hydrophobic. And also, the R group are going to influence the shape of the catalytic sites of enzymes. For those who took A-level, they know this very well, that the activity of the enzyme depends on the catalytic site and the shape of the catalytic site. And this catalytic site can be influenced by pH. And the pH is very important because if you change the pH, then the catalytic site, which has an R group, can be either protonated or deprotonated. So if you change the charge on the R, the interactions between the different R will change, and therefore the shape of the protein will change. Don't worry, this information you're going to become more familiar with it when we study the proteins lecture. Now we come to the last part, which is the classification. So we classify amino acid according to the properties of the R group. So we have hydrophobic amino acid, hydrophilic neutral, hydrophilic with a negative charge, and hydrophilic with a positive charge. So these are the 4 classes of amino acids. The first one is the hydrophobic. And again, to classify them, what do you look at? The R group. If the R group has only C and H, they are hydrocarbon. Remember, when you have only hydrocarbon, they are not capable of forming any type of bond with water. Therefore, they are going to be what? Hydrophobic. Okay, so look at the R here on the square. The R has only C and H. Therefore, these are the hydrophobic amino acids. But actually, here also they don't have a benzene ring, so we call them non-polar aliphatic side chain. Aliphatic means non-aromatic. We have the second part of the hydrophobic, the one that are aromatic, and these are the 3 aromatic: phenylalanine, tyrosine, tryptophan. All 3 are hydrophobic. Some textbooks classify tyrosine as hydrophilic. So that's why in a test you don't expect from me to test you about tyrosine, because there is disagreement whether tyrosine is actually hydrophobic or hydrophilic. Then we have the hydrophilic, which are neutral. So they are friend with water, but they don't have a charge. But what makes them friend with water? They can form what? Hydrogen bonds. And why do they form hydrogen bonds? Remember our previous lecture on hydrogen bonds? Either because they have OH or SH or C double bond O, or they have an NH. So all these are capable of forming hydrogen bonds. Therefore, these amino acids are classified as hydrophilic. There is also disagreement on methionine, because they say they have a sulfur and the sulfur can participate in hydrogen bond, but some textbooks they say no, the sulfur is hidden. So again, methionine case is like tyrosine, no agreement on whether it's hydrophilic or hydrophobic. The last 2 are the hydrophilic with a negative charge. So not only they can form, they dissociate in water, but they can form ions. And these ions have negative charge, okay? Or we call them basic or acidic. If they form a negative charge, is it a basic or acidic? Acidic, because they donate the plus, the H plus, so they will become negative charge. And these are aspartate glutamate. And 3, histidine, arginine, lysine, they are hydrophilic but positively charged. So these are also classified as what? Acidic or basic? Basic, because they will accept the H plus and they will become positively charged. This is a practice question for you. This is the kind of question you should expect in a TBL or a midterm. Which one of the following statements concerning the peptide shown below is correct? The peptide contains glutamine. Does it contain glutamine? No, there is no GLN. The peptide contains a side chain with secondary amino group. No, because what? No proline. The peptide contains the majority of amino acid with a side chain that would be positively charged at pH 7. What are the ones that will be positively charged? Will H, will L, will A? Do we have these? Histidine, lysine, arginine? No, we only have arginine, so there is no majority. The peptide is able to form internal disulfide bond. What do we need to have internal? We need to have 2 cysteine. Do we have 2 cysteine? Yes. So the correct answer is D. Okay, this is the suggested resource for practicing question. Lippincott's flashcards for biochemistry. And this is an example of one of the flashcards. So it has all these practice questions that you could solve them. I'm not going to go through them now for the sake of time. And this is what we have at the end of each chapter. You have a concept map. This is very important because it summarizes all the information that we have studied in the lecture. Thank you for your attention. And sorry about the time delay because of the technical issue. Yasser, can you come and take the attendance, please? If you have questions, you can come to my office. One announcement about my office. I'm located on the 3rd floor. For girls, you can come at any time. For boys, you can only come after 1. Okay, boys are not allowed to go to the female area except for after 1 o'clock.